Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
Invisible to the naked eye, they account for the majority of Earth's biomass. For at least 3.5 billion years, they have dominated the evolution of life. So diverse in form and distribution, they are Earth's most ubiquitous life forms, carving out niches in ecosystems ranging from the vastness of the open sea to the digestive tract of a termite. And yet, only 1% of them are known to science. They are the microbes, and without them, life as we experience it would soon come to an end. In fact, it would never have begun at all. We are not alone. You can't see them, you can't hear them, you can't even feel them. But no matter what you do, no matter where you go, they are with you. Tiny, intimate strangers, the microbes. The average human being is composed of 10 trillion cells. An impressive figure, yes? Well, ponder this. For every cell in your body, you harbor 10 times as many microbes. That's 100 trillion microscopic tenants on and in your body. And if the thought of 100 trillion microbes inhabiting your body is making you squirm, don't worry. Without microbes, you wouldn't last a week. From a microbe's perspective, we're just walking, talking ecosystems. For as long as human beings have been around, we and the microbes have been evolving inextricably together. I'm Jim Leinfelder. I'll serve as your guide over the course of this 12-part series, during which you'll be introduced to a category of life that is still largely unexplored. And you'll meet the scientific pioneers who are exploring it. You've all heard the expression, seeing is believing. And that's certainly the case in microbiology. True awareness of the widespread distribution of microorganisms and some of their characteristics didn't occur until the development of the very first microscopes. Microbiology, as the name implies, quite literally focuses on organisms too small to study with the unaided eye. They are protozoa, algae, fungi, bacteria, and with the invention of the electron microscope, viruses. But how small is small? This is the tip of an ordinary straight pin. Under an electron microscope, we see it first magnified 30 times. But then, as each image increases in magnification by a factor of 5, we can see increasingly close-up populations of rod-shaped bacteria clinging to the scored sides of the pin's point. And viruses are many times smaller than this. Though the organisms microbiologists study are extremely small, the scope of microbiology is vast, touching virtually every aspect of human existence. In agriculture, microorganisms help break down waste and aid in the fixation of vital nitrogen in the soil that would otherwise come from chemical fertilizers. Microbes are used to treat sewage, clean up industrial waste, and decompose the garbage in landfills. We use them to make food such as cheese and bread. And yet, we do our best to eradicate them from our kitchens. There are bacteria in our large intestine that synthesize vitamins, allowing them to be absorbed into the bloodstream. And herbivores, who depend on cellulose as their major food source, can't digest it on their own. Only the bacteria in their stomach have the enzymes needed to break it down. Cows, sheep, and other herbivores get their energy from the byproduct of microbes. A very tiny minority of microbes act as pathogens or disease-causing agents, representing a natural threat not only to humans, but to all life forms. We therefore attempt to control potentially harmful microorganisms to prevent infection, especially in environments such as hospitals. But it wasn't so long ago that we lacked even the basic understanding of how to control microbes before contamination and infection got a foothold. The results of our ignorance were often dire. Most of recorded history is dictated by disease. During the American Civil War, for example, more soldiers died of infection than of any other cause. Thousands were wounded, allowing microbes into the body, resulting in infectious disease. Because effective control measures were unknown, doctors had few ways to prevent or treat these infections. The microbes took over, the body couldn't defend itself, and the person often died. Delivering mothers of the time faced the same threat. Deaths from infectious diseases acquired during or soon after childbirth were as high as 25%. As the relationship between microbes and disease became clear, control methods were developed, such as the concept of personal hygiene, washing with clean water and soap, and improved public health measures such as a safe water supply and sanitation. Disinfectants, antiseptics, and most importantly, vaccines were developed, and soon we were increasingly effective at preventing infection and disease. However, once infection occurred, we still lacked life-saving treatment. In the early 20th century, doctors could do little more than prescribe isolation and rest for people suffering with diseases such as tuberculosis and many others. There were no cures. Then, in 1928, British chemist Alexander Fleming made a startling discovery. Fleming noticed that a mold he was growing in his laboratory killed encroaching bacteria. The mold was Penicillium chrysogenum, and it produces the antibiotic that we call penicillin. Although we credit Fleming with the discovery of antibiotics, the fact is microbes invented them billions of years ago.
Industrial monument to the miracle drug. Mass production penicillin plant at Terre Haute, Indiana, one of many where the life-saving medicine is now being manufactured wholesale. Tons of mold like that you've seen on spoiled foods are processed by rapid fermentation.
In the 1940s, penicillin became available to the general public. Diseases that had claimed hundreds of thousands of lives for centuries, from pneumonia to syphilis, could now be cured. Today, we can prevent and treat diseases more successfully than at any other time in human history. We still, however, have much to learn about the relationship between microorganisms and humans. Tragically, infectious diseases still devastate human populations worldwide, most often in places where sanitary public health standards are not or cannot be practiced, and appropriate medical care is not available. And there has been an explosion in the number of bacteria resistant to antibiotics, and an alarming but inevitable emergence of new life-threatening microorganisms. But while this lethal minority get most of the media attention, without their less celebrated legions of beneficial relatives, the threat of disease would be a moot point, because life itself would be impossible. It was the microbes that made this planet a viable place for all other life forms. Although plants are generally credited with developing the ability to convert the sun's rays to food energy, photosynthesis, it was actually ancient microorganisms, the cyanobacteria, that first produced oxygen as a byproduct of photosynthesis 1 billion years before the first plants ever existed. Over the next 3.5 billion years, microbes evolved to fill every niche, creating an environment in which plants, animals, and humans thrive. Microorganisms now maintain balance in the environment by playing a key role in the recycling of vital nutrients such as nitrogen, sulfur, phosphorus, carbon, and oxygen. Today, all over the world, plants depend on microorganisms to feed them nutrients. In turn, the plants supply 50% of the oxygen we breathe. The rest comes from marine microbes, especially algae and cyanobacteria. A visible form of algae is seaweed. Like plants, algae are photosynthetic and help to anchor the food web in the ocean. Marine bacteria and algae help maintain the world's supply of oxygen by playing an essential role in the carbon cycle. Primary producers, such as algae and plants, draw energy from sunlight, taking in carbon dioxide and releasing oxygen. Consumers, such as animals and many microorganisms, use oxygen and give off carbon dioxide. It's an endless cycle, and only one of the countless ways microorganisms have supported life throughout our planet's history. Without this cycling of nutrients, the surface of the Earth would be as barren as the moon. The invisible microbial world is rich in its diversity. It is populated by more different kinds of organisms than all of the visible world combined. The diversity of all life is derived from the microscopic crucible of life, starting with the first microbes. Microbes reproduce quickly. Their generations fly by. And because they've been around for billions of years, they've had the opportunity to shuffle and reshuffle every possible combination of their genetic information, their DNA. As the environment changes, so do the organisms, which in turn can change the environment. As scientists have begun studying DNA, a molecule shared by virtually every living creature, they've discovered a method of comparing the ancient pedigrees of organisms to look back in time and unlock the mystery of how all life is related. This technology is also being used in a myriad of ways, from mapping the human genome to developing new antibiotics. But microbiology is still a very young science. We actually know very little about most of the Earth's microbial tenants. Scientists across the globe are searching for these unknown microorganisms and their role in the subsistence of life. The first step in this search is their discovery, collection, and identification, a step we have barely begun to take. Marine microbiologist Steve Giovannoni is combing the Sargasso Sea for microbial life that will help us understand how life works in the world's oceans. A generation ago, scientists failed to appreciate the vast diversity of marine microbes.
There were plenty of skeptics. There were people that thought that the kinds of bacteria that we already knew about were a perfectly good example of what lived in the oceans.
Giovannoni and his team have returned to Oregon State University to look for undiscovered microbes among the samples of water from the Sargasso Sea. But how do you know when you've found a new microbe anyway? Searching with a microscope is not realistic because so many different types look the same. Imagine this is Giovannoni's mystery microbe, unrecognized in a sample of the Sargasso water. When he looks, he can't identify his quarry simply by physical appearance.
We had to find some other way of identifying them. And what we did was to become gene hunters. In place of the organisms, we began to hunt their DNA.
Giovannoni chemically broke open millions of different microbes. The messy results were billions of different DNA fragments, all mixed together. DNA is a complex genetic recipe that consists of two strands held together by four chemicals called nucleotides, which are like puzzle pieces. The length and arrangement of these four chemicals determines the identity of every living thing on Earth.
We removed the DNA from the microorganisms in the seawater, and we took single genes. We took single copies of genes from all that DNA, and we studied those single genes. And we did it by reading the code on the gene.
Giovannoni retrieved pieces of a specific gene, the one coding for ribosomal RNA. Scientists used this gene to identify organisms. He compared the ribosomal RNA with an up-to-date database of existing codes, the genetic codes of all known living things. One by one, the codes matched existing organisms. But one code did not match up. It belonged to an unknown organism. They had discovered the existence of the mystery microbe. They named it SAR11, Sargasso gene number 11. Now the real work would begin. He had to find the whole microbe. It wasn't long before the search took on greater significance. Giovannoni's research team found SAR11 in water everywhere they looked. And they knew it was a bacterioplankton, which means it is involved in some way with the supply of nutrients to algae, the oxygen producers of the sea. Following Giovannoni's lead, other scientists found the same gene in ocean samples from around the world. Evidence even turned up in freshwater lakes. Even more remarkable, in every sample of water, there were more SAR11 genes than genes from any other organism. Giovannoni realized that he was on the trail of possibly one of the most abundant life forms on the planet.
Right now, all we know about SAR11 is one gene, and we know that that gene is out there in the sea.
Stephanie Conan is a graduate student in Giovannoni's laboratory.
We feel that it plays an important role in the food web in the ocean, and we would like to find out what that role is.
But so far, they hadn't managed to grow SAR-11. And for over a century, the only way to study microbes was to grow them.
This is one of the oldest methods in microbiology. It's called a spread plate. And really all it is dropping water from the environment on the surface of a sterile plate that contains food for bacteria, separating the bacteria so that they grow up by themselves and form colonies that we can see with our eyes.
1 week later, so many bacteria have grown that they become visible. For years, microbiologists believed that the colonies which grew like this on spread plates revealed the majority of the microbial species on the planet. The reality was different. Over 99% of all microbes on Earth will not grow this way.
The real challenge today is to try to grow SAR-11. Something about these cells makes them unable to grow in a scientific laboratory, at least today, using the kinds of techniques that scientists have. I told Stephanie this, and I said, what would be really a great discovery, but challenging and require creative thought, would be to try to isolate these organisms for the first time. Do you want to do that project? And she said, yes.
We are going to be testing different food sources for this organism to determine what it eats. And if we can do that, we can start determining what its role is in the food web.
Our hypothesis is that SAR-11 eats organic carbon, but what we don't know is what kind of organic carbon does it eat? What is it like to eat? And that's one of the things Stephanie has to figure out in order to make it grow.
If Stephanie's diet for SAR-11 does get it to eat, grow, and reproduce, they will be able to find out much more about what the microbe actually does. Unlike many microbes, SAR-11 thrives with very little nutrients, so Stephanie is tempting it with very small amounts of food, 1,000 times less than normal. But it's a stubborn microbe.
We think that the reason it's so difficult to grow in the laboratory is that possibly SAR-11 is a small cell built to live at the very low nutrient concentrations that are found in the open ocean sites that we study, and that it's precisely adapted to do one thing, and if we change anything, then it doesn't divide in the lab. So the trick for us will be to somehow or another reproduce exactly what's happening out in the ocean in order to make it grow.
Although she has not yet been able to get SAR-11 to grow, Stephanie's persistence paid off in a different way. She used SAR-11's single gene to get the mystery microbe to reveal itself and pose for a photograph.
Under the best microscopes, SAR-11 is nothing more than a pinpoint of light. It's like looking up at night and seeing a star in the sky. And they're a lot smaller than most of the other bacteria out there. In many cases, they're 1,000 times smaller. You could put 1,000,000 billion of them into a tablespoon. What's it doing out there?
What is SAR-11 doing out there? That's a question that applies to the vast majority of microbes on the planet. We know they have great power, playing paramount roles in disease and its treatment, in industry, public health, and in the Earth's evolution and very functioning. But we know so little about so many microbes. To know more, scientists are studying the molecule from which all the functions and diversity of life is derived, DNA. In the next episode, we'll explore how DNA found in all living things is expressed in the same fundamental structures of all life.
Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
For information about this and other Annenberg Media programs, call 1-800-LEARNER and visit us at www.learner.org.